Is the second in a mini-series on our soil mapping and research. Last time, Sam and Linda introduced the Smap Soils Information System, and today we'll talk about the new research on water retention in soils, the new soil moisture estimates in Smap, and their impact on some key models. If you would like to remind yourself about the earlier webinar, or if you missed it, you'll find a link to all previous webinars in the follow-up email that you get today. At the end of today's session, Sam and Linda will also provide a sneak preview of some new work on farm-scale soil mapping. We'll talk for about 20 minutes and leave time for questions at the end. And we will do our best to get to all of the questions if we can. But if we don't answer, rest assured, we will follow up with you directly. Responses to all questions will be posted with the webinar on our website in the future. Just to help me out, put your questions in as early as you can. And without further ado, over to you, Linda and Sam.
Great, thank you, Christine. So this webinar is about the new soil water retention information that we will soon be releasing on Smap. What is soil water retention? When we have, it's really important, it's about the capacity of soils to retain water. So when you have the irrigation water or rainfall and the water, the soil is full of water and there's excess water, we call that saturation, as you can see on that first diagram. Once that excess water is gone and you end up with the pore space being full of water that's then available to the plants, we call that field capacity. Once all that water is gone, it's either drained or it's been taken up by the plants, we call that wilting point. Available water is the difference between field capacity and wilting point. If you look at the next diagram, you can see that that wilting point and field capacity, they vary according to the soil type. And so too does the difference, the available water. It varies with the soil type. So that information, that available water, is really important for information about how well the crop will grow, for irrigation demand, for nutrient losses. Now, this research that we've been doing, it's part of an MB Endeavour Research Fund. So that's a program called the Next Gen Smap. It's a 5-year program. And we're now in the final year of that program. So this research is actually a culmination of the last 3 to 4 years worth of research. And in this webinar, we're going to talk about 2 approaches to estimating those key soil properties, the soil water retention properties. We're going to look at first a national approach, and then Sam's going to come in and talk about the farm approach. So starting with the national approach, and many of you may have seen this slide before. We've used it in many Smap presentations, and it's just illustrating that we've got the underlying data within Smap. Then there's the modelling, where we turn that underlying data into information, which we then deliver through a variety of platforms. This webinar will be focusing on the first 2 layers, starting with the data and then the modelling and extrapolation of that information. So what is the new additional data? The laboratory data is a mix of new and legacy data sources. So we have gone out into New Zealand and done a whole lot more sampling and sent those data samples off to the laboratory to get these new measurements of the soil water retention. We've also dug deep into the old literature, old Excel files, old laboratory cards to retrieve data that was measured long ago. And we've retrieved that and then added that into the National Soils Database repository. Clearly, we've had to do quite a lot of QA on particularly the legacy data, and that's been quite a big process. But the net result is that we now have a significant increase in the amount of data that we have that we can then use to develop our models on. So the existing model that's on Smap Online at the moment was developed back in 2014. It's actually 6 years old. And the new model we're referring to as the 2020 model. And so back then in 2014, we had 313 sites that were used to develop the model. That's now more than doubled, and we've got 684. Each site, there are a number of layers within it, and each of those becomes a sample. So we've gone from 1,400 samples up to 4,600. So it's a really big jump in the data. The lines here, so we can, there's a New Zealand soil classification. We've just divvied it up by that classification so that we can see where the points are. This middle column here indicates the area of each of these classifications within SMAP. And so the bigger ones are in bold. And so we've doubled and tripled the amount of information in the browns and the palex, also in the recents and the pumices. But note that the pumices do have a smaller number of sites. And so the data is less representative of the pumices, but of the browns and some of these other soils, there's really good representation. Another way of looking at the representation is to think about it in terms of the texture. So this is the silt, sand, clay texture diagram. What we have here is the red contours indicate the distribution with texture of the measured data. The black points are the SMAP soils. So clearly what you want is to have overlap, and so that the measured data is a good representation of the soils that are out there in New Zealand. And you can see that with the right-hand one, with the recent, there is in fact a good match between them. This is not the case with the palex. You can see here, so this is high silt. There's a lot of measured data with the high silt, but no measured data where there is high sand content. So this becomes an extrapolation exercise. We're needing to take what data we have here and extrapolate out to and this is what we've put quite a lot of attention on in this model development, is checking the extrapolation that it is in fact valid. This paper here was written a couple of years ago, and it documents that development of the model. We tested 3 different types of model and decided on one of them, and this is the relationship for predicting that soil water retention curve, the wilting point, the field capacity, total porosity, from which we can then derive available water. This is some of the results. So in this, I haven't put in all of the orders here, just some of the key ones. Green, the green points are the results of the new model, so the 2020 model. Orange are the results from the 2014 model, and the black points are the measured data. And you can see generally the black points are quite widely distributed, and in fact the green points are capturing that wider distribution better than the orange ones. But let's go and have a look at that palette that we were just talking about here. So there you can see, so it's silt along the x-axis. We can see lots of measured data here in the middle, but no measured data down here where there's high sand, which is what I showed you in the earlier graph. So that's the extrapolation. But in fact, those palettes, they tend to be in Canterbury, the high sand ones tend to be the lower gravelly layers within the soil. And actually, pedologically, they're really similar to the recent soils with high sand. And you can see up here that we do have quite a lot of data with the high sand content. So that's how we're able to do the extrapolation in this case, is we use the recent data to help us make the predictions for the palette high sand soils. Another one that's interesting to look at is the pumices. So you can see here that with the high sand, the green points, the new model, are a bit lower than the orange ones, and that matches the black measured data. At the other end of the scale, where we have high silt, the green points are actually got increased water content, reflecting again that measured data. Let's look at some of the differences between the 2 models. The 2014 model is based on logistic regression. The 2020 model, and this goes back to that paper where we trialed 3 different types of formulations for the model and determined that the beta regression was the best format. We checked that again with the 2020 modelling, and it is indeed still the best form for our model. There's an additional input parameter from the 2014 model, and that's using the depth down into down the profile of a given estimate or prediction that needs to be made. And another change is the peat layers. Now they're quite different. They're not actually part of the beta regression model. What we did for them in 2014 was we had some default values for those key parameters, and now with more data we've been able to change those default values to be more representative. Now, I've already talked a little bit about the increase in data from the 313 up to the 684, and we've got that increase in the number of samples. But because we've got more samples, we also have more samples that we can train the model with, but also more samples that we can then validate it. So we've gone from 280 samples up to 928 in the new model. In 2014, we certainly had very low site numbers and samples for some of the soil orders. We still have that for some soil orders, but others have increased by 2- to 4-fold. Now, one of the bits of research that we've done in the last few years has been done by a PhD student at Lincoln. And he has been investigating whether stones hold water that the plants can then access. In the 2014 model, the assumption was that they didn't hold any water at all. Whereas what he has found that in fact the grey wacky stones do hold a small amount of water. And so that information has now fed into the 2020 modelling. In 2014, very limited error information, whereas now we have a full error model for all of the points that are being estimated on that water retention curve. We can access those for the 2020 model. But the really big difference comes in the extrapolation. So within the training data, we can get the goodness of fit statistics between the 2014 and the 2020, and actually they're much the same. But the difference is in the extrapolation. So what was happening in the 2014 model was that there were 424, so 10% of the siblings. We actually couldn't predict those key properties that we feed into Overseer because the extrapolation was too unreliable. Whereas now it's just 4. So with a much bigger reference data set, we have a more stable model and the extrapolation is more reliable. And that's the really big sort of change and improvement. Well, let's sort of start aggregating that up to profile available water. So that's the available water in the top 60 centimetres. What have been the changes there? So let's look at the table first of all. So total, this is the median change over all of the siblings in millimetres. It's a slight decrease of 4.5 millimetres. And you can see the change by some of the key soil orders where we have the most soils within New Zealand. The browns haven't changed much at all, just 1 millimetre on the median less. The elephantix down by 10 millimetres, the pumice is down by 23, the recent's down by 8. But that's just the median. What you really need to have a look at is the distribution of changes. So if you move over to the right-hand plot, the box plot, so the boxes indicate 50% of the values and then the outliers beyond that. And so the 0 lines down there through the middle. And so if it's below, like the glaze, like the pumices, like the recents, they have mostly decreased in value in their PAW from the 2014 to the 2020 modelling. Whereas some semi-arids and granulars have increased. The ones that are close to the 0, like the browns, haven't. They've got a little bit above, a little bit below, not a lot of change. What does that mean spatially? Let's look at the South Island first. So the colour scheme here is green. That is no change, or up to 5 millimetres, so not much. The blues are an increase in PAW, the profile available water. The oranges and yellows are a decrease in the PAW. And so in Canterbury, there's not a lot of change, a little bit more of an increase. Down here in Southland, on the other hand, there is a decrease in the PAW. And that's because they've got clay soils down there, which have decreased a wee bit. A little bit different in the North Island. You can see a lot more of that red and orange and yellow where the PAW values have decreased. And that's because of the pumices here and the elephantix here have decreased in their PAW. Some increase over here in Hawke's Bay. What does all that mean in terms of the models that we then feed this information into? So John Bright from Equilink has very kindly done an analysis where he's taken 8 locations around the country, and then we took a 1 kilometre radius and looked at the soils within that radius. He's run that through Equilink and then put them onto this plot for us. On the x-axis here, we have the percent change in PAW, whereas the y-axis is the commensurate change in water irrigation demand. The numbers here are 10% here, 10 plus 10% there. So really it's within plus or minus 10%, that's where most of the predictions have fallen, with a couple of exceptions, one down there and a couple of up there. A wee bit of a different story for the overseer results, where there's a bit more change. Now what they did was they took 32,000 farm analyses from across New Zealand and ran them through overseer, looking at the before and after the change in terms of the nutrient losses. The vast majority of farms, so 71%, so this is the less than 10% change, plus or minus, 71% sit in there. 54%, which is this group here, are getting an increase of some sort. 8%, this group here, that increase is more than 20%. And then there are some that, in fact, have a decrease in their nutrient losses. In terms of greenhouse gases, nearly all of it is within that plus or minus 10%. So there certainly are some significant changes in some areas of New Zealand that will result as come about as a result of the changes in SMAP. Now, Overseer are clear that they will not auto-update the budgets for these new soil values, but if any new request is made for SMAP data from Overseer, then it will pull in the new information once we do the release, which is later in August. At that time in August, all of our delivery platforms will be updated at the same time. So what I've done there is describe the national model to you. Sam's now going to give you a preview of the farm-based approach.
Thanks, Linda. So yeah, one of our challenges that we've faced for a number of years now is how do we use SMAP and the modelling power and the rich data that Linda's just given you an overview about to support consultants or farmers that want to be able to do more detailed work at the farm scale. And one of the challenges of a national model is it's a global prediction. In other words, it's aiming to predict across all the variety of soils across New Zealand. So there's inherent compromises within that model so it can cover all of that variation. And so the other limitation there, which has been a real challenge, is the variety of input parameters that you can put in. We know that there is attributes such as soil carbon or bulk density, that if we could get access to that at the national scale, we don't have data for that at the moment, or predictions of that would greatly improve the model. So there's a real opportunity there if we could develop a method by which we could collect that information at the farm scale, use the power of SMAP, then we could potentially be able to downscale SMAP to be used at the farm scale. So we've got a vision here which is around developing a method in which a range of consultants who may have limited soil expertise, they don't have to be a pedologist like me, but they could be able to cost effectively collect samples and characterize a broad range of those soil profile attributes, those input attributes I talked about, at that farm or paddock scale. So move to the next slide, Linda. So what we've just worked on is a proof of concept project supported by the Fertiliser Association, Ravensdown and Balance as well there, to pick up on some research that we've been working with a lot of international colleagues that's rapidly advancing in recent years around vis and mid-infrared sensing. And this is really where pedology is going and soil survey is going on a global scale, and we've been investing in that, and it's got real potential. So if you look at the bottom on the left-hand side there, you've got a farm map from one of our case study farms that we did there, and what that shows is that the consultant has mapped out the pattern of soils on that farm, but they're really quite limited in terms of how they characterize attributes in a quantitative manner to be able to provide key inputs into tools. So here, for example, you can provide a description of the soil and the soil photo, but that doesn't necessarily help for a lot of the modern tools which are out there, which require quantitative attributes, measurements of carbon, measurements of water holding capacity, sand, silt, clay. And so what we've tested here is a workflow by which those consultants could use a statistical sampling scheme to randomly sample those different areas of soil types there. And you'll see just above the map, there's a range of techniques by which you could do that. You could use a hand auger, or likewise, both within New Zealand and internationally, there's a lot more mechanical sampling devices that you can use to collect samples now. And what that gives you is a core down the profile. You can see Linda's cursor there has moved over to that, which samples the whole soil down to whatever depth you're interested in. In the case of overseer, it'll be to 60 centimetres. And then you can measure that in increments, so 10 centimetre increments is what we measured it in, and use the vis or mid-infrared scanning. The real advantage of the vis or mid-infrared scanning is from that one scan, that spectral scan that from that sample, you can then predict a whole suite of attributes. And so that's where we've also been doing work, building on that in the National Soils Database, which Linda introduced at the start, and the samples that we have sitting alongside that, we're processing and trying to scan those to build up a spectral calibration set of equations for a suite of attributes that are down there. And building that into the Smap modeling engine, and from that we can then use the power of the Smap modeling engine together with those farm-specific information to be able to customize the soil information, the soil fact sheets that we provide out, and likewise through to tools such as Overseer. So that was a quick introduction of the Smap farm test. But really, in terms of key messages from today's talk, you can see from today that Smap is science-driven, which means that science evolves, science moves on, our knowledge does develop. So Smap has always changed and always will change. We try our best to manage those, which is partly today's talk, and make sure we communicate that out to people and work with people as best as we can. And the real aim is to keep improving the information that we're supplying. Soil information is crucial to a whole range of land management policies, decisions, and as well as from farm scale up to the national kind of scale. We have been very lucky in that we've had a significant investment through MB, our MB research program, which has really allowed us to focus on the soil water data over these last 4 years. And that has made a step change in terms of the reference data that we've been able to use to develop our models. And so the outcome from that has been the new national model, and that is a significant improvement in terms of the robustness of that model, which Linda has gone through, in predicting soil water retention values. It's important to recognise it's a national scale model, so we're predicting across all of the variability of soils in New Zealand, so that will mean that all of the soil moisture values associated with those soils will change when you use a national model. We're using the we've also been working on a prototype Smap farm test, so how we can downscale Smap to the farm test, and it's showing real potential for applying Smap science at the farm scale, and we've been looking for a method such as this for a number of years now, so we're actually starting to hone down into something which is workable now. We've presented that proof of concept there, and our next stage is we're currently in the process of trying to put together a partnership to progress this to the second stage, so it can actually go out as a functional service to people. All of this really is reliant on a collaborative funding relationship across a whole range of partners. So we really do want to be able to acknowledge the support of MB, MPI, a whole suite of regional councils, the Fertiliser Association, and also a lot of farmers across New Zealand, which over these last 4 years have continue to allow us to access their farms and be able to dig pits and measure the attributes we're using now to improve Smap. So that's the end of our talk today, and we'll hand back to